Hi. Well, we're starting the afternoon session. Thanks for being here after lunch. I know that it's going to be a little bit difficult, but just a small reminder before we start with the talk. Remember that there is a bag missing, a bag of the event, one of these bags that you received during the registration. There is a bag missing that belonged to Professor Christefan. And the problem is that there was a charger inside the bag. So if someone of you found this bag or took it by mistake, please let us know. We don't care about the bag, but the charger is a little bit more important. So with that, let's start with the talk. It's a pleasure to introduce Professor Juan Claudio Toledo. He's a professor at the Institute of Nuclear Sciences of the University, and then he is going to talk about a topic that I think is of interest to all of us. Thank you, Juan Claudio.
Thank you, Max. So this is good. Yeah, thank you. Good afternoon. So I'm going to talk about some work we have done here at C3 about climate change, but in the perspective of complex systems. We've analyzed data, climate data, by taking concepts that we have found or learned rather in the study of complex systems and then apply these ideas to data from climate change. And we think that we have found a very interesting result about how the regulation of the climatic system has changed in the 20th century. It goes hand in hand, of course, with everything that has been said about climate change recently. So I'm going to start very basically by discussing how that is important to us in the field of complexity research, right? So one of the driving questions that we have is this: how do complexity and organization emerge from simple interactions, right? Complexity science is much about understanding how we make this jump from simple elements and simple interactions to systems which are called complex, that have emergent properties that are difficult to predict from the interactions of the elements, right? So this is the essence of complex systems. And we're interested in these systems because in the real world, many systems exhibit this property of having complex emergent behavior, which is difficult to predict from the simple interaction. So of course, in biology, which is perhaps the most important example, that we are really unable to fully understand how life occurs from the basic laws of chemistry and physics, but also economy, sociology, or other fields of science, which are called today not the soft science, maybe. These are not the hard science because it's difficult to make predictions in these fields, right? And this is because of this reason, of the fact that it's very difficult to predict these complex behaviors from the elements and the interaction of the elements. So there are many aspects to this question, or rather many ways in which a system can be complex, can exhibit complex behavior. One of them is emergent structure or behavior. I think I lost this. Okay. Can you hear me? Yes, you can, right? So one of the things that emerge is structure, right? So we can have complex structures like the patterns of fractals emerging from very simple mathematical rules, right? And the whole structure that emerges from these rules is very, very rich, very deep, very, very complex, very complicated to study. And yet the rules that make up this pattern can be very simple. We also have self-organization, which is a very important topic in complexity science, which is spontaneous coordination of a system of agents, of a system of interacting elements. And this coordination can emerge even without a central coordinator, so to speak, right? So just because of the simple rules of interaction of these elements, then we have complex organizational behavior in the system. We also have adaptability, which is a capacity for a system to adapt to changes in the environment. Of course, life is perhaps the greatest example of this that we know in the universe, but also there are other systems, non-biological, that have the capacity to adapt to changes, to be resilient against perturbation or stimuli. And finally, many of these properties really are there because we have nonlinear dynamics in these systems, right? So, of course, chaos, these are two examples. Here's this turbulence, of course, in a climatic systems, which is difficult to predict because turbulence is chaotic, it is very complex, and also we have chaos in the more traditional sense of studying mathematical systems that exhibit the property. So the point here is that very small perturbations at one point in time in the system have a very long-lasting effect on the dynamics of the system, and so therefore it's hard to predict the behavior in the future, right? So this is a consequence of nonlinear dynamics. So complex systems have all of these properties, right? It's what they have in common. It's very difficult to define what complexity is, a bit in the same sense that it's complicated to define what life is, right? But at least these properties seem to be common to all these systems. And in particular, one of the things that we have studied here at C3 in I think, a good deal of detail is phase transitions, because many of us are physicists and we studied these phase transitions when we studied physics. So the concept is very simple, of course, right? It's when a system has a certain dynamics, a certain way in which it is behaving, and then something happens, something changes, and the dynamics, the behavior changes radically. So for instance, this is an example of a classical problem which has a phase transition, which is the problem of percolation. So if we imagine this, I have this, right? If we imagine a grid of spaces, and the black spaces are spaces that are blocked, and then we put water on the top of the grid, right? And the question is whether the water can find a certain path towards the bottom of the grid, right? And so what we can do is just grids at random with a certain probability of space being filled or not, being blocked or not. And here's an example for 40% probability, then 60%, then 80% probability that a given square will be blocked, well, will be free versus blocked. And what we can see is that if there are enough free spaces in the grid, then the water will percolate through the grid. So there will be a path that connects the top of the grid to the bottom of the grid, right? And the important point about this is that as a function of this probability, right, of how many sites are open or vacant in the grid, the probability that the system will percolate has a sudden transition, right? So at low probability, at low site vacancy probability, the system almost never percolates. There could be some, just by chance, a given grid at low density that can percolate, but it's very unlikely. And then as we reach this value, 0.593, then suddenly the percolation probability jumps almost up to 1. And at any point above this probability, then the system will almost always percolate. So this is an example of, conceptually at least, an example of a phase transition, of an abrupt change in how a system behaves. It's very quick. It occurs in a very narrow range of probability here on the x-axis, and it completely changes, in this case, the most important aspect of the behavior of the system, right? So this is a phase transition. Now, why is it interesting? Of course, this point here is called the critical point, right? The point at which the transition occurs, which has a specific value in this case of the probability of the vacancy of the sites, is called a critical point. So why are we interested in these critical points? Is because not only the transition occurs there, but many interesting properties emerge at this point precisely in the system. Some of these are that the correlation length scales increase. So if I try, not in the percolation system, but in general, if I try to measure the correlation between 2 points, two places or 2 agents in the system, in general, the correlation will increase, the correlation length. That is, how far does a perturbation has an impact in the system will increase when we are near or at a critical point. We also have, what I just said, the appearance of long-range interactions, even when the interactions in the system are short-range. Even if the system is made of pieces that only interact locally, at a critical point, we have very long-range interaction between these elements. We also have the propagation of perturbations that decays more slowly, and this can be perturbation in general, or can be information in particular. So think of the brain, right? perhaps at a critical point, in a critical way, the brain is organized so that information can travel more efficiently towards many different parts of the brain. It can connect different areas of the brain more efficiently if the brain were critical, which we still don't know. And also we have scale invariance and other like more mathematical properties of the system that we can measure, right? So scale invariance, power law scalings in some of their properties, fractal structure, etc. So the point is that critical points, these lie often at a sort of boundary between ordered phase, which is like, no, it has a structure, very ordered, and a phase that is disorganized, that is more chaotic, more stochastic in some sense. So in a way, critical points are a sort of dynamical balance between these two extrema, okay? Okay, so a system we have studied a lot here at C3 is the Ising model. The Ising model is a very old model in physics that was initially created to study magnetism in materials, right? So there's a phenomenon that was discovered at the, I think, the end of the 19th century, where you have a magnetic material. If you change the temperature of the material high enough, there will be a point in which the material stops being magnetic. It will lose its capacity to retain a magnetic field, right? That's called the Curie point in physics. So there was a very simple model that was created to study this property. How does a magnetic material loses its capacity to become magnetized? And really, in a more general sense, this is just a model of information and interaction of things. It goes much beyond just magnetic fields. So it's a model of how local interactions can produce global behavior that is interesting. And in particular, the point is that the Ising model has a phase transition, a very well-known, well-studied phase transition. So let me just briefly discuss how this works. We imagine that we have this lattice of spin sites. Each of these arrows is a little site in the magnetic material that can be aligned either up or down, right? So the two arrows represent the two possible orientations of each spin. And so the point is that whether the grid has a global magnetic field or not depends on the balance of these two orientations. When they're about 50-50, then the magnetic fields of each side sort of cancel with those of the other orientation, and there is no magnetic field globally, right? So the plate is not magnetized. On the other hand, when the orientations are aligned with one another, then we have a magnetization globally, right? So it's a very simple system, a very simple way to explain magnetization in a material. So we can study this because we understand, we can write very simple equations for the dynamics of the system. This is the energy of a site, which is simply defined as the product of the spin state, which is either -1 or +1, multiplied by the spin states of the neighbors. So the point is that this is a model of just local interaction, right? The energy at one point in the grid depends only on the relation between the point in question and the neighboring sites, right? So the energy is defined just locally. And the point is that physical systems in general, what they tend to do over time is to minimize the energy, right? So this will translate into a rule for how this spin site in the center will behave in time. So if initially we have this state, which if we plugged in here, right, the values of the site, of the spin sites, this is a high energy state. So over time, what it will tend to do, it will want to transition into the spin up orientation, right? Because in this configuration, this site has a lower energy. So the point is that one of the key properties of the system, of the dynamics, is that over time, the spin sites want to align with their neighbors, right? That's what they want to do naturally. If you leave them be, they will try to align with their neighbors. So this is the first component of the Ising system. The other component is that we imagine that this plate, this magnetic substance, is immersed in a thermal bath. So it is subject to a temperature of the environment, and this temperature will induce stochastic fluctuation in the spins. So it will try to randomize the orientation of a spin site, of every spin site. And there's a rule that comes from statistical mechanics that relates the change of energy to the probability of there being a change, a flip of the spin, and this depends on temperature. So as temperature of the bath increases, then the probability of this thermal flip of this random flip will increase, okay? So this is the second component of the Ising system. And the point is that these two effects, they compete with one another, right? One effect wants to align the spin sites, and the other effect, the other part of the dynamics, wants to randomly change them. The system will become ordered or not depends on who wins, whether the natural tendency of alignment wins or the stochastic fluctuations win. And the point is that there is a temperature, a critical temperature at which these two effects are in balance. At that point, we'll have a critical point, we'll have a phase transition. So it's a point of dynamical balance between two competing dynamics in the case of the Ising system. So we can plot here the total global magnetization of the lattice versus temperature. And what we see is that what happens is exactly what they observed in real magnetic materials, that at low temperatures, the material can become magnetized easily. It can retain a very strong magnetization. But as we increase the temperature, there is a point, critical temperature, at which the capacity to become magnetized becomes completely lost. And then the magnetic field disappears, and there is no magnetization globally, right? So this is a phase transition for the Ising model. So the point is that, as I said, these critical points are very interesting in many ways. So what we can do with a system like this, a theoretical system like this, is try to measure quantities from it. So for instance, we can study the statistics of time series that we extract from the system. We can, for instance, look at the global magnetization of the grid, and then see how this evolves in time for a given temperature, and study the statistics of the signal. So we can do this to Monte Carlo simulations of the model in order to have an evolution in time of this, and we study this time series of magnetization. And the point is to study how the fluctuations of this time series in time, how are they? What are the statistics? So what happens at the critical point with these things? So here is an animation of the computer simulation of the model. Here, the two spin orientations are represented by dark and bright dots. And here you can see the evolution of the magnetization of the system, right? As a function of the number of generations that we have simulated. So initially, there's a transient in which magnetization will simply slowly increase, because as you see here, one of the two orientations will win. In this case, the bright orientation wins, and eventually almost all of these islands of dark orientation will die, and then the time series will stabilize just below 1 here, right? This happens at low temperature, when the temperature is too low for the random fluctuations to be important, right? So what dominates here is order, is just the dynamics, the tendency of spin size to align with their neighbors. Then we can go to high temperature, the other extreme of the temperature range. This is, again, a simulation of this. And you can see, even if we started the simulation with all spin sites aligned in the same direction, very quickly, the spin sites will randomize, and eventually we'll just have fluctuations around 0, right? So in this grid, around 50% of the sites are in each of the two orientations at a given time. Of course, this fluctuates because it is a stochastic system, so it will just fluctuate around 0 over time, right? And really interesting part is what happens when the temperature is exactly the critical temperature. So this is a simulation of this. And here, the grid is a little bit small, but what you can see here is that there are these islands of likewise oriented spin sites that sort of appear and disappear, right, over time. If we let this evolve, the magnetization will never stabilize towards any equilibrium value. It will just go all over the place from 0 in terms of magnetization. And here, if we assess the size of these islands, we'll find that there is a very nice power law that describes how many islands there are of any given size, right? So this has a sort of fractal nature, which is not very obvious here because the simulation is a bit small, but if we made a very big zoom out, we would see that the structure is the same at every scale, right? So because of the distribution of the sizes of the islands. So at a glance, this becomes a really interesting system because it is fractal in structure. But what we wanted to do is study the statistical properties of the time series of the system. So as I said, we take the magnetization time series, what I showed here, this time series, and we study some of the statistical properties of the series. So one of the first things that come out is that if we study the variance of the time series, that is how much it varies, how much it fluctuates in time, we can plot this variance of the time series versus the temperature of the system. And what you can see is that at low or high temperatures, the variance is very small because there's an equilibrium point at any given temperature other than here, and the time series will simply fluctuate around this equilibrium point with very small fluctuations. But when the critical point is approached, the variance just explodes, right? So the variability of the time series becomes much, much greater. And this is very important because this has been observed in climatic time series already a couple of decades ago, right? So it's a sort of hallmark of a phase transition, the fact that the variance will explode near a critical point, in the vicinity of a phase transition. We can also look at other statistical moments of the time series. For instance, we can look at the skewness, which is a measure of how symmetric the distribution of fluctuations is. Are there more fluctuations to the above a given value, the mean, or below the mean? And what we can see is that at low and high temperatures, the skewness is relatively low. At high temperatures, it's basically zero. At low temperatures, it's not zero, but not very high. And as we approach the critical point, this become more asymmetric. So this is a reflection of the fact that the statistics, the distribution of the fluctuations change as we reach the critical point. At low or high temperatures, actually, it's sort of near Gaussian, and at the critical point, it becomes something completely different. This is the kurtosis, which is a measure of the likelihood of extreme events in the system. And again, a kurtosis of 3 indicates the value for a Gaussian, so the normal amount of extreme events. And as you see, when the critical point is approached, the kurtosis changes abruptly. So for instance, here we have many more extreme events that we would expect in a Gaussian distribution. Again, this is important in climate change because one of the consequences of climate change is that there are more now extreme events in weather in local parts of the world, right? Okay, so and finally, when we can also study about the time series is how much correlation it contains. Autocorrelation, right? Are the values in the future dependent on the values of the past of the time series? So that's a question of how much correlation it has. And one way to study the correlation, not the only way, but one way to study the series is by studying the power spectrum of the time series, which is simply a mathematical trick that we do in order to more clearly see these correlations. And the important point is that when we plot this power spectrum, it can look in one of these ways, but not the only way, but these 3 ways are special ways in which the correlations can look. So for instance, when we have a time series that is random, which is called white noise, the power spectrum is flat. So this means that the energy of the time series is distributed equally among all scales, all frequencies. The power spectrum, by the way, is just the power versus the frequency, okay, of the Fourier transform of time series. So for white we have that spectrum, and then for these 2 kinds of correlated noises, we have a power law spectra. That is, we have these straight lines in a log-log diagram, and the inclination of the line is a measure of how much correlation there is in the time series, okay? So 1/f noise is a sort of interesting, like middle-of-the-road kind of noise that appears in many places in nature, actually. And Brownian noise is simply like a random walk, right? This is a statistic of someone walking and having little displacements with respect to the previous position. So the point of this is that the power spectrum is a measure of correlation, and that when we have power loss in the power spectrum, the inclination of the power loss is a measure of how much correlation the time series has, okay? So what we can do is I'm showing here the power spectra of the Ising model. So the temperature is changing. It's here in the upper right corner. So as you can see, let it loop again. At low temperature, it's very flat. And as we approach the critical temperature, it becomes a very nice power law all across the whole range of frequencies that are plotted here, right? So the conclusion from this is that the correlations at high or low temperature are like the correlations of white noise. There are no correlations or very small amount of correlations. And when at the critical point, the correlations become much stronger. and the correlation length becomes much larger. So that's what we can conclude from the fact that it's a very nice power law across all the frequencies. So what will we learn from the study of phase transitions in a system like this? That scale invariance, that is the fact that we have power law correlations in the power spectrum, occurs at critical points. We also have found that 1 over F noise, usually found in real systems, and we think that this is an indication of the stability of the system, but there's a bit of controversy about this idea. And also that as a phase transition approaches, then the time series offer very nice indicators that the phase transition is approaching. So we can study the statistics of time series of the system, and then this can tell us whether we are approaching critical point. This will be very important. And finally, it seems that these properties have a certain amount of universality. That is, we can find this behavior in many different kinds of systems, in percolation, in easing systems, also in many other kinds of complex systems, and even in real systems, or we think that that is the case. Okay. So the second point I want to talk about, this was phase transitions and why they're important to study the dynamics of a system. The second point is talking about this very nice model, theoretical model also, that is called Daisy World. This is a model of self-regulation, right? So it's a very simple model that was developed in the 80s by Lovelock and Watson in order to study how life could regulate the conditions of a planetary environment, right? Because as you know, the atmosphere of the Earth is completely changed by the presence of life. The oxygen here comes from bacteria a long time ago. So the conditions, the atmospheric conditions of the Earth depend on the presence of life. And these guys thought that maybe there's a sort of feedback pattern between life and physical properties of a planet that can make life more successful on the surface. Okay, so the model is also rather simple. We imagine a planet that is inhabited by 2 kinds of daisies, white daisies and black daisies. The difference between these two is that white daisies are hardly reflective. They reflect sunlight, right? So the energy that comes from the sun towards the planet is reflected back into space by white daisies. And the black daisies have a low reflectivity. So much of the energy that comes to the sun of this planet becomes absorbed by the planet and thus by the atmosphere, right? And the point is that the temperature of the planet is determined basically by how many white and black daisies there are at any given time. When you have more white daisies, the planet will be cooler because more sunlight will be reflected back into space. When there are more black daisies, it will be much hotter in that case. And an important point is that the daisies have a preferred temperature. Here around 22 degrees Celsius in the model, this is the growth curve of the daisies. So they grow more efficiently around 22 degrees, and they die if the temperature is below 5 degrees or above 40 degrees Celsius. Okay, so there is only a range of temperature in which the daisies can live, and there is an optimum for the daisies, the temperature that they prefer. Okay, so the system is governed by a couple of differential equations that are shown here. I won't go into too much detail. This is the areas, the fraction of the area of the planet that is occupied by white and black daisies. So this changes time according to this differential equation. Each equation has a growth term, which is this. This is the growth function, which depends on temperature. And this is a death rate. So they naturally die after a certain time, and this is quantified by this symbol here. And the point is that we can solve these equations numerically. Apart from these two, we also need to express how temperature of the planet is related to the albedo. This alpha is the albedo, that is the percent of light reflected back into space. So as a function of the albedo of the planet, the temperature of the planet, simply by an energy balance equation, right? So by coupling these equations, we can solve in time for the areas of the planet that are occupied by each of the 2 kinds of daisies. And here is the important result, okay? This is the temperature of the planet here in the y-axis, and this is the luminosity of the star. That is how much energy is being received from the star on the surface of the planet, right? And here, focus first on this black dashed curve. This is simply what we would get from physics, right? As the sun becomes more luminous, there is more radiation, the temperature of the planet increases. This is a very simple thermodynamic result, right? Which is what we would expect in a planet without life. But then comes life. The blue curve here is what we get with the presence of daisies. So what you can see is that even when the sun is relatively cool or not very luminous, there is a point where suddenly the temperature jumps abruptly. And here the green line is the preferred temperature of the daisies. So the point is that there is a point here, a given luminosity of the star, at which daisies can, for the first time, live on the planet, and they will distribute over the planet. The whites and the blacks will distribute in such a way that the temperature of the planet will increase very near their ideal value of temperature, right? So this is an example of regulation. The fact that daisies are there and that they can die and reproduce means that the system will adapt in order to try to regulate the temperature around this sweet spot, right? And then, as the temperature of the planet is increased, one would expect an increase of the temperature. I mean, the luminosity of the star is increased. One would expect an increase in temperature, but this does not happen. Because as the luminosity increases, the daisies will redistribute. The black ones will become less dominant, and the white ones will become more dominant in such a way that the albedo will be changed such that the temperature remains almost constant, right? Constant and near their preferred value. And then if we keep increasing how much energy is produced by the star, there is a point in which it's too hot for them to live and they all die, and then we return to this thermodynamic equilibrium curve, right? So this is an example of self-regulation, of how without any rules, the daisies don't know anything about temperature, about sunlight, about reflectivity, right? All they do is simply respond to the environmental conditions of the planet. And as a consequence of this very simple behavior, the temperature becomes regulated. It becomes very stable despite a very large increase in how much energy is output by the star, okay? So this is the key point of the model of daisy world. It's a model of self-regulation, okay? This shows the populations of the two types of daisies versus luminosity again. So the blacks are this green curve, and the whites are this blue curve. And this explains how they achieve regulation, right? Which is what I said. When the luminosity is very low, so the planet is very cool, almost all daisies are white, because these are those, black, because these are those that will absorb sunlight more efficiently. And as the temperature, the luminosity is increased and the temperature of the planet is increased, the white ones will start to become more dominant, right? So at this point, there's only white daisies remaining on the planet, because the blacks would make the temperature too high for them to live. So this is how they adapt to the system, to the change. Okay, so daisy world is a sort of old model. It's a 0D model in the sense that we only have two numbers that describe the state of the system. But what you can do is make the system more interesting, more complex, in order to study phase transitions in a biggest detail. So what we have done is simulate a 2D version of this, in which instead of just having two numbers, how the area of the planet that is white and black, now we have a lattice, and each of the sites in this lattice has either white or black daisies, and there is a rule for how the lattice evolves. And together with this, we have a heat transport also simulated with the system. So this is a heat transport equation. We have diffusion here. We have emission and absorption in these two terms. And now we consider that the reflectivity of the daisies is continuous in 0. Being black or white daisies, there is a continuum between black and white. So there are gray daisies of varying levels of gray. And this is simulated as a cellular automaton, okay? I'm going to show a couple of simulations. This is the temperature of the planet in a 2D simulation. This is the white spots are spots barren without daisies, and this is the albedo of the surface, which is of course determined by the color of the daisies, which goes between 0 and 1. Okay, so as the simulation evolves, here we see a 3-time series. The red one is the temperature of the planet. The blue one is the albedo. The albedo, and the green one is the vegetation index. So the point is that the temperature starts at a certain point and then quickly descends and then sort of stabilizes around a given value. Here, 22 degrees is about the temperature that they prefer, right? But as you can see, that there's a lot of fluctuations. You can also see it here, right? How there are patches of high temperature, patches here of black daisies that then disappear, and then white daisies appear, or intermediately colored daisies appear. So there are a lot of fluctuations in the time series of temperature in the system. So in this 2D case, more complex case, the regulation is even stronger. This is, again, planet versus the luminosity of the star, and the blue curve is what we get in the daisy world simulation. So it's very, very stable. The temperature remains almost constant across this big range of luminosities, right? So in 2D, with these rules, with diffusion in temperature, it's even better. The regulation is much stronger. So what we can do is also study the time series statistics. So this is the power spectrum of the daisy world model for a temperature time series. So as we can see, the luminosity is here. You will see that in the mid-range of luminosity, there is a very clear power law, like we saw in the case of the Ising model. And as luminosity becomes too high, there starts being this kind of elbow, of knee in the power spectrum. See, this is regulated. This sequence is a regulated planet, and then as we reach about 1.1, 1.2, regulation is lost, and the power spectrum becomes curved here. You see? There we lost the regulation. And one thing that we can do with this model is study how greenhouse gases or the greenhouse effect change the regulation. That is, if we introduce a greenhouse effect in daisy world, can the daisy still regulate the temperature of the planet? And the answer is that yes. What we do is in the heat transport equation, we add this term here in red, where G is a sort of index of how strong the greenhouse effect is. So if it's 0, it means that there is no greenhouse effect, and we can increase it up to 1, right? And what we get in the case of this daisy world with a greenhouse effect, here is the temperature of the planet in red and the albedo in blue. And this, in the x-axis, is the greenhouse forcing G, right? So 0 means no greenhouse forcing, so there's no greenhouse effect. And then we increase the greenhouse forcing and see what happens. So amazingly, the daisies do well, right? This is the temperature of the planet again, and it doesn't matter if we start increasing the greenhouse effect towards 1, the temperature remains the same, right? How they do this? By changing the albedo again. So daisies will exist, will live on the planet, so that the albedo will become higher, and so this will compensate for the greenhouse effect, which is the heat in the atmosphere of the planet. This is true up till this point, about 0.4 in the greenhouse forcing. This is completely destroyed. At that point, all the daisies die. The temperature becomes huge, becomes very large, because there's no regulation anymore. And well, the albedo becomes whatever, because they're all dead, right? So this is the point at which regulation is lost. As you can see, it's a phase transition, in the sense that everything is fine, 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 up till this very point in which everything goes to hell, in this case, right? All the daisies die, life is over on the planet. So regulation is very strong. It's a very, very nice effect, a very resilient effect, but it has a limit, okay? And well, these are the statistical properties of the time series. This is the variance standard deviation of this time series. As you can see, it's about constant with some fluctuations, right? And here, at the point where regulation is finally destroyed, there is a peak of variance. Like we saw in the Ising model, the variance increases near a critical point. And the same thing happens with the skewness, where for any of the 2 time series, we have a very abrupt change, right? The temperature skewness is here, and then it goes here at the loss of regulation. And likewise for albedo, the skewness is around here, and it jumps up to here, right? So there are very important changes in the statistics of the time series as this point of no return is crossed, okay? Kurtosis, same thing, right? Very stable, around 3, around Gaussian, and then it becomes huge at the moment when regulation is lost. Okay, So to summarize, Daisy World, it's a model of self-regulation. The stability of the planet is characterized by power law spectra, as we saw in the case of Ising. This happens at critical points. The system is resilient, that is, we can change, we can increase the greenhouse effect, and the system will remain stable. And then when the capacity to self-regulate is lost, there are very marked changes in the statistics of the system. So we can know that the system is changing in an irreversible way in this case. So it's a very powerful illustration of how a biosphere can adapt to the environment and how it can shape the physical environment in which it lives, right? But the point is to remember what happens when we push it too far. So finally, climate change, okay? How am I doing in time?
Doing well.
Yeah, I'm doing well? Okay. So finally, climate change. This is the center of all the work, right? The previous was just ways in which we learn how these systems behave in general in a very theoretical way. So now we want to look at the real data. So this is the global temperature of the Earth. It's not measured in absolute temperature, but rather in what scientists call anomaly, which is simply how much above or below a given temperature that was standardized the temperature is. So 0 is that this is the reference temperature, and this is higher, this is lower, right? And that is arbitrary reference. This is time for the past 1,000 years. So as you can see, this is called the famous hockey stick curve. It looks like a hockey stick, right? So the temperature was relatively stable for the last 1,000 of years, and then from about 1 century, 2 centuries before today, it started increasing, right? This, now we know that is caused by anthropogenic activity, in particular by industrial activity in the world, okay? This is the uncertainty of the measurements. It's big, it's big.
So you don't know if you have...
It's true. But the statistics of these things have been very, very studied. So we have a very good idea that the averages are about right. The problem is here in the not so distant past, there were no thermometers registering temperature, right? So we have to rebuild, to infer the temperature from other kinds of things, right? In the red curve, which has almost no uncertainty, this is direct measurement of the temperature, right, in the modern era. I'm going to talk a little bit about this. So how do we measure the temperature, at least in the geologic sense? We can use ice cores from Antarctica, so these big cylinders of ice, and what they do is trap isotopes of oxygen, and we can infer a temperature of the distant past by looking at how much of this isotope was present there. We can also look at tree rings because the growth of trees depends on the temperature of the ambient. We can look at coral growth with the same reason. The temperature makes coral grow more efficiently. And we can also look at sediments in lakes and look at, again, the isotope content of these things. So this is the temperature record for the past many thousands of years. So this is the present here. The graph is up like in reverse. So this is the present. This is years. So as you can see, in red is the temperature, and in blue is the CO2 content of the atmosphere. And there was a very big downward trend over the past 1,000 of years. And then this very stable era began, which is called the Holocene. And many people think that animals and human beings were able to succeed on planet Earth because temperature became very stable. We're not very sure why this happened, but there's a very clear indication that this happened about 10 to 12,000 years ago, right? So the temperature has become very stable since then on the Earth. Now, we now have direct temperature measurements, right, from 200 years ago to the present approximately. So this is an example, but there's a problem with this. This is an example of the temperature in New York City. I don't remember which year this was, but I think it was recent. So as you can see that the temperature, it's hard to read the numbers, but this is about 20 degrees Fahrenheit, I think it is. And this is about 90 degrees Fahrenheit. So as you can see, the temperature will change, right, during the year. These are the months of the year. It will change by a huge amount, right, from 20 here of 30 degrees Fahrenheit to up to 80 or 90 degrees Fahrenheit. So this big range of temperatures is a problem because if I go back quickly to this to this plot, just look at the vertical scale, right? This is about the average temperature for the past 1,000 years, and then there is an increase of about 0.6 or even 0.5 degrees Celsius, right? So climate, global of around 1 degree Celsius of increase. And then at a given point on the Earth, we have a wide variability of the temperature during the day for once, right? Night and day, and also because of the seasons. So measuring this very small 1 degree effect is very hard. But luckily, many groups have took this challenge and done very, very complicated and well done work in order to try to measure this very small variation. So this involves trying to isolate the rest of the effects from just the global warming. So what they do, what they produce from this is what they call the global average temperature index, okay? So this is the global temperature, again plotted with respect to an arbitrary reference for the past about 200 and something years. As you can see, there's much variability here because we didn't have many measurements from back then. But as the 20th century entered and many more stations around the world start recording temperature, and then all this data can be collected and analyzed together, then the uncertainty, which is the shading here, becomes rather small. And here I show 4 different groups, which are intergovernmental groups, which are international coalitions in many cases of scientists to make the construction of a temperature, and they all sort of agree, right, that around the second half of the 20th century, the temperature started increasing almost linearly, right? And now, today, we're around 1 degree, almost 1 degree of temperature of average. Now, this may not look big, 1 degree, but keep in mind that this is the temperature of the whole atmosphere, of the whole planet, right? So this represents a very large amount of extra energy that is being retained by the atmosphere. So it's a very big thing, and it has many possible consequences. For instance, this big amount of energy that is retained now will make the polar ice, both in the north and the south, melt more during the respective summers. So this means more water towards the oceans. This changes the currents inside the oceans. This redirects currents that transport heat from the poles to the equator and vice versa. So this can have a very big effect on the planet, okay? This is, again, just the last about 35 years of the record, where you can see how these groups really agree about the global temperature, right? So they have different analysis techniques in order to isolate just the global temperature, and they agree to a very high degree. So I think it's very that global warming is happening, and of course, and that it's made by man. The reason why I think it's anthropogenic, that is made by man, is because we know from physics that if you pump CO2, carbon dioxide, into the atmosphere, the temperature will increase. It's a very well understood effect. And here is the paleologic CO2 record. As you can see, that there have been periodic variations in the CO2 concentration in the atmosphere that, in fact, are correlated with ice ages. But if you look at the scale here, this is in fact, this plot is a bit old. The current value is above 400 parts per million, okay? So just in the last 50 years, we have already doubled the amount of CO2 in the atmosphere. And we know from physics that putting CO2 increases the temperature, so there's no mystery that's happening and who is causing it, okay? All right, so this is just to summarize the fact that the global temperature is rising. This is a real effect. Scientists are not discussing this anymore. It's agreed upon. And that the cause is anthropogenic, so it's caused by man. There is no doubt about any of these two things. Don't listen to whatever orange-haired politicians have to say about this, right? So finally, I'm going to go a little bit faster. The point of the work that we did is to see whether by studying the statistics of the time series of the temperature record, we can see any of these trademark behaviors that occur near phase transitions, right? Because that's the hypothesis, right? that there has been a certain equilibrium in the global temperature of the Earth, and the pumping of CO2, the forcing that we're doing, is changing this equilibrium, okay? So we have analyzed one of these temperature reconstructions, the one for the Berkeley group, which is this one. It shows, again, a couple of hundred years, and the red one is the average. So this is the global warming, basically, in the second part of the 20th century. And here we're looking at power spectra of this global temperature record, okay? So it's going decade by decade. We have partitioned the record in 10-year intervals. And I cannot stop this because the computer is there, but let me see if the next one, yeah. And the point that we see is that in a certain part of the spectrum, there is a flattening of the spectrum, okay? You will see that the later decades seem to be, these are the first decades, right? 1910, 1920. And then as we go towards the present, you will see a flattening. It's not very evident here. So what we can do is, with analysis, we can measure this. Okay, so this is the oldest decade in the record and the more recent one, which is complete. As you can see, there are many different parts of the spectrum. I didn't go into much detail, but the spectrum has different dynamical sections. So this high frequency part is like the local dynamics of the weather. And this more intermediate scale has to do with the scale at which transport, both in the oceans and the atmosphere, can redistribute energy across the globe. So this is more, this part is the one relevant for the global dynamics, okay? This is more like local weather. And the point is that the oldest decade, if you compare here these two parts, it's much flatter at these scales, at the global scales at the present, okay? And also, it's a little bit steeper at the high frequency parts. So if we try to measure how flat these power laws are, we get these two plots. So this red one is the one for the mid-scale, the one relevant for global transport of energy, and this is the one for local fluctuations, so the weather, right? And even though there's a lot of variation, as you saw, the spectrum is kind of noisy, it's hard to really fit a line, but we've done a careful statistical study of this by using surrogate time series and resampling of the data. And the point is that even though there's a bit of fluctuation, there is definitely a downward trend. So this is a downward trend of the correlations present in a time series. So the correlations are being lost at these scales. There was a certain amount of correlation back in the start of the 20th century, and as time has evolved, this has been decreasing, right? The error bars are from statistical analysis. This is the autocorrelation function. I won't go into detail about this, but this plot here, when I'm showing the lag 1 autocorrelation, which is a traditional indicator of phase transitions also, is like monotonically increasing almost during the 20th century. This is the spatial distribution of temperature, which we have also analyzed. I'm going to skip this and just show this is the variability of the spatial fluctuations of the Earth. There's an upward trend here. We can also study the skewness, which has a small upward trend here in the last part of the time series. And kurtosis, this is an artifact of the data, but the point is that this is not, I think. So there's also a departure from the value of 3, which indicates Gaussian statistics. This is the top 1% and high temperature and low temperatures. And you can see that the high temperatures have been slowly increasing. So this is like the extreme events, right? An extreme event of temperature on the Earth, where 1% of the temperature is this high. So also there's an upward trend of this. So an increased likelihood of extreme events. So to conclude, we see that scale invariance of the power law of the spectrum seems to be broken with the 20th century, that spatial variance increased steadily, and also that the other moments also exhibit trends that suggest or tell us that a phase transition may be approaching, a critical point may be approaching. So, okay, so that's it. Thank you.
Are there questions, comments? No?
Yeah.
Thank you, Claudio. Very nice. I would like to know if you have analyzed precipitation information, because also this is very important in order to have periods with no rain and things like that.
Yes, of course. We haven't, because this was focused on like global average parameters. No, so I guess we could try to measure the global average precipitation, but I think precipitation is more of a local phenomenon. Now it depends on the orography of the geography of the site, the proximity to water, etc. Meanwhile, temperature is more of a physical parameter that just depends on the balance of energy between the sun and the earth. So it's a sort of a more global parameter, which is why we have focused on this one. But we could start looking at more local things to see, for instance, if there has been more droughts recently than before, more extremes in how much rain falls in different parts of the world. Yes.
Denis?
The spatial DAISY model, how noise is introduced there? Because you've presented an equation.
Yes.
But how
it's gone.
The noise and the stochasticity, how is it?
Because the way the grid, the lattice of sites evolves, is controlled by a cellular automaton which has probability transitions. So whether a daisy will propagate to a neighboring site is determined by a probability. So basically the program just throws a die and decides whether this happens or not. So that introduces the stochastic part of the model, because the daisy world, the original model, doesn't have a stochastic part.
And have you attempted to compare climatic data to the prediction of this model?
Daisy world?
Yeah.
Yeah.
So daisy world regarding the slopes and the close to this critical time or point that are.
Yes, we don't think that daisy world can be used as an actual model of the Earth, because the Earth is much more complicated as a physical and biological system, right? But the point is just to understand what happens in very simple models like daisy world when we break regulation capacity, or when a tipping point is approached, and then try to like make the analogy with what we see on the data of the actual Earth. So using daisy world directly, no, but learning how transitions happen from daisy world and then transporting that learning to what we see on the climatic data, that is what we have done.
I was wondering if there is maybe a dialogue among the scientific community about the names we use, and talking about the orange fellow, you know.
Yes.
Global warming sound really bad, you know. So, for example, we have this super storm coming from the north of United States.
Yes.
And the reasoning is, where's the global warming? You know? So I don't know if it's just a curiosity if among scientists there is maybe a proposed change to a more realistic thing like global disequilibrium or something.
Yeah, I guess that global warming is a very specific thing, is the increase of average temperature. And then climate change is a broader concept, which is the consequences of this increase in temperature, right? So for instance, the frequency of strong storms in the Atlantic will be one of the aspects of climate change, right? So, and there's evidence recently that the storms are getting perhaps not more frequent or bigger, but they're growing at a higher rate, which is called intensifications in meteorology. So this is happening much more faster than it used to be, right? So these are aspects of climate change.
One last question.
Yes?
Here?
Yes.
Well, actually, also correlated question. There's this phenomena, which I actually forget the name of, which is the wind current, the air current around the world. And one of the explanations of about why we suddenly have also extreme cold temperatures is about these currents that come
Yes.
from the Arctic and start affecting the weather.
Yes.
My question is, do you have or happen to know a relation, the exact relation on why when the temperature, the global temperature increases, these air currents start to flow more around the globe?
Well, I'm not sure. We'll have to ask an atmospheric scientist about that. But there will be an interrelation between transports of energy in general that mediate. This is the jet stream, I think, what you're talking about? The jet stream in the upper atmosphere? Or no? I'm not sure. Yeah, but I don't know. We'll have to ask an atmospheric scientist. Yes.
La mía no. ¿Eh?
Sí, sí, sí.
Okay, we're starting. I think this is the last talk of the evening session. And before this, we have a cocktail and the art exhibit. So it is a pleasure for me to introduce Professor Rafael Peña Miller. He's a professor at the Center of Genomic Sciences of the University in Cuernavaca. And now we're changing the topic to talk about bacteria again. So you have seen that this is there's a variety of different topics here. So please, Rafael, thank you for coming.
On the contrary, many thanks for the invitation. So the work I'm going to be presenting today is basically or mostly work done by my students, my graduate students, in particular, Charlie, Bruno, and Raul. And also my collaborators, Ayari Fuentes from the Center for Genomic Sciences, and also in Spain, the groups of Álvaro San Millán and Craig McLean at the University of Oxford. So conventional wisdom states that if you have a bacterial cell like this one, in this case, this is Salmonella, the human pathogen, and then it grows and it divides and it produces another cell, and we assume there are no mutations, no genetic changes during that process, then these two cells are, they're genetically identical, but it is believed, or it has been believed for decades, or it was believed for decades, that these cells are also essentially, for all practical purposes, identical. They live in the same environment, they have the same genetic background, they are essentially the same cell. In this case, we have fluorescently tagged a virulence gene in Salmonella, and you can see as you continue watching how the colony grows, that some cells turn on and off this virulence gene. So this is called phenotypic heterogeneity, and it's been research for the last, 10, 20 years, and is one of the main drivers in my research questions in my group. I'm not going to talk about Salmonella, although I wish I was. I'm going to talk about something a little bit more fundamental in a way. And it has to do with, which is probably the fundamental problem in systems biology, which is how do you map the genotype to the phenotype? I once said this in front of proper biologists, and they got really mad at me because this is a question of biology, not of systems biology. I'm sure they're right. But it is also a fundamental question in systems biology. And in particular, if you think it from a systems biology perspective, there are two things that drive this process, and one of them is the signal, the deterministic component of the system, which it is a complex dynamics that emerges from nonlinear interactions between genes and many things that interact in multiple temporal and spatial scales. It's a very complex thing. But also it is, of course, subjected to stochastic forcing. So you have extrinsic and intrinsic noise. It comes from everywhere. Transcription is inherently stochastic, but you also have many, processes in the cell are basically just occurring with very small copy numbers. So the occurrence of those reactions is usually a stochastic process, and therefore there's noise everywhere. Of course, noise also comes from outside. You also have asymmetric division. If you have low protein numbers, for instance, I don't know, some proteins, key proteins in the cell, I don't know, there are like 7 in a cell. So the moment the cell divides randomly, the cell divides, then these proteins can randomly distribute between daughter cells, and that can induce some sort of disturbance into the system. I think that's what we're gonna look into a little bit more in this talk. So in general, the questions that we're trying to answer or address in this talk is how do genetic perturbations propagate through this cascade of noisy and complex cascade and express in a phenotype. This is, of course, not a recent question, and it's definitely, this is just, I was just playing around. This is a very old question, and maybe one of the first, it predates systems biology for sure, but it was addressed by, it was an interesting question back in the 1940s when, this is before the DNA structure was discovered. So, this was really before or in the beginnings of molecular biology. And Salvador Luria and Max Delbruck realized that, or asked the questions, are mutations spontaneous or induced? Actually, the question was not asked by them. It was a question that everybody was studying back then. And what people were trying to do were what's called fluctuation assays. So it basically just do not put any selective pressure, just let the population drift, divide, and grow. And then at some point, you introduce selective media, for instance, an antibiotic, or in the case of Luria and Delbruck, it was a virus, a bacteriophage. And then you see how many of them survive. So some of them must have acquired a resistance mutation, allow them to survive this novel environment. The question was, is it something that mutation, or that's something that allowed them to survive, was it something that occurred in response to the selective agent, or was it something that was already there because it occurred at some point before the selective agent was induced? And many people were trying to do these fluctuation assays, and they realized that they were very difficult to study. And these are the two scenarios. So if it's an induced one, here the blue ones survive. If it's an induced mutation, then when you see the selective pressure, then that's when some of the cells acquire the mutation and they survive. Well, if it's spontaneous, it could have occurred way before the selective agent was used, and therefore you would expect a higher number of surviving populations, or lower number. It depends. And that was the whole point. There was, if you do this experiment, and they're very easy to do and very interesting, you realize that if you count how many cells survive and you repeat that experiment multiple times, you find that the variance of surviving cells, of the distribution of the cell count, it has a huge variance. There's a lot of variability. And back then people used to think that variability was something to do with the experimental system. And the brilliance of Luria and Delbruck was to realize that that was not an error, but it was actually a feature of the system. And that they will have very elegant statistical arguments to show that precisely that variance suggests that this hypothesis must not be true, and then the only possibility that you have is that they are spontaneous. So this is a very old question, but there are, of course, and a very elegant explanation. There are, of course, assumptions within their model, and one of them is that what they're assuming that to be a mutation is something that is phenotypically mutant. Well, that's what they're measuring. They're just only measuring how many cells did they survive. So they're not really tracking when did the mutation occur. They're actually tracking how many of the cells already are in the mutant type that allows them to survive. And basically, they're assuming that this map, the genotype-phenotype map, is instantaneous. No? The moment you have a mutation at the genotypic level, you will express at the phenotypic level as well. And last year, that was 60, 70 years later, in Sebastian Bonhoeffer's group, they repeated the same experiment, and they introduced, they allowed, put in a batch culture just cells to grow, and then put some selective media, and counted how many colonies did they survive. And then they repeated the same experiment, but before using the selective media, they actually propagated a few more times in non-selective media. So they were postponing selection. And when they did that, they observed that you will have many more, or considerably more, mutants presenting. They refer to this process as phenotypic delay. And in this particular case, it has to do with a chromosome, how it replicates, and it produces something that's called effective polyploidy. When we read this thing, we formed ourselves a hypothesis. Of course, we have been working on this for long before, but we never really thought about this. And this is what I'm going to try to talk to you today. If this is true, then if you are carrying the gene in multiple copies within a cell, then, for instance, multi-copy plasmids, I'll explain what that means, you will increase phenotypic delay. And by doing so, you will provide an evolutionary advantage for cells in fluctuating environments. So that's the plan. So first, what do we mean by plasmids? About plasmids. Plasmids are small DNA molecules that can replicate independent to the chromosome. They're very important, and people have been studying them for a long time because they can either be important from a bioengineering perspective, but also because they can, in natural environments, they can carry beneficial genes, either from social behavior like nitrogen fixation, but also, and critically, for drug resistance. And the reason they're very interesting from an evolutionary perspective is because they are also the responsible for a process in microbiology that is called conjugation, which is when two cells basically share horizontally through plasmids genetic material. You can imagine that this is a very important thing to do. I mean, not only transmitting vertically, but also transmitting genes horizontally. Plasmids are inherited. I'll go a little bit further because that's actually a very interesting point. How are they inherited? What we have been focusing in this line of research is to study multi-copy plasmids and see how they can accelerate the rate of adaptation, elevate evolutionary trade-offs, and also increase intercellular genetic diversity. There are some papers if you want to read more about them. Anyway, I'll carry on. So then about segregation of plasmids, or how do they replicate? First, our experimental system, and every data I'm going to present here today is E. coli, standard E. coli, and a plasmid that is a very small plasmid, but it's carried in multiple copies in the cell. It has around 20 copies per cell. It doesn't have this possibility of moving horizontally. I mean, it doesn't have the metabolic, the machinery to do conjugation. So it's basically just stuck in one cell. And we're also going to put fluorescent markers so we can see when a cell is carrying a plasmid. We can see through microscopy and image processing. And we're interested in seeing how selection acts on this plasmid. So we also introduce a resistance gene. It's like 5,000 base pairs, so it's very small. Yeah, some plasmids are way larger. Some plasmids are as large as a chromosome in some cases, and those are usually very complex. They have active partitioning systems to make sure that one cell carries one copy, and they have also toxin antitoxin systems. I mean, there's a whole zoo of plasmids. This is in the spectrum of the very small. So in multiple plasmids, and doesn't have any of those fancy molecular machineries. So how do they replicate, or how do they divide? They transmit towards the next generation. So the moment a cell divides, it divides at the middle, I don't know. And then because plasmids, and in particular small plasmids, don't have this active partitioning system, then that means that they're uniformly distributed randomly distributed within the cell. So the moment the cell divides, some of the plasmids are with one of the daughters, and the other plasmids are with the other daughter cell. And so in this cartoon example, we have 5 plasmids. This one inherited 3, this one inherited 2. And then they have a control mechanism, a copy number control mechanism that basically means that they start replicating plasmids every time throughout the cell cycle. And when they reach the mean copy number, whatever it is, they basically stop replicating. The molecular mechanisms of this are very well studied. And then it goes on. Of course, there's a probability, and that's an interesting feature. We'll talk about that later, but there's a probability of the daughter cell not inheriting any of the plasmids. No? And in that case, of course, there's no regulation or no replication. So the first thing that I want to point out is that this has to produce, I mean, you have something that increases and then decreases and increases and decreases. I mean, there's like a, you replicate and then, so they have a, it's an interesting mechanism. So they have their own kind of like antitoxin system. So they stop replication when the concentration of that thing is above, it's high. But then as the cell starts growing, it starts diluting this thing, and then it forms regions within the cell that are very low concentration of these things, and then it starts replicating. Basically, they reach some sort of carrying capacity. And then they segregate, and then they replicate, and then they segregate. So this is a noisy process, of course, and then it has to produce, in this case, because we can see the fluorescence, I mean, we're using a fluorescent marker. You can see in this picture that you have a distribution of fluorescent intensities, which may or not may be due to plasmid copy number variability. I mean, in a population, you can count and you can say like, oh, there are 19 copies in average. But from a perspective of a single cell, you're all the time varying the number of plasmids. So how do we model this? So there are two things, segregation and regulation. Segregation, as I mentioned, it is basically just random and it occurs upon division, and the probability of plasmid loss is given by a bimodal distribution. It's a Bernoulli trial. Replication, on the other hand, is proportional to the difference between the expected mean copy number and the current number of copies. So if you have very little copies, it's more likely that you'll replicate that plasmid. Well, you replicate this plasmid. It's basically, from a probabilistic point of view, it's like an urn model. No? You take out one plasmid and you replicate that one and put it again. So you have two stochastic processes. Like you can think of them from a probabilistic point of view. And they have to produce, well, they're stochastic in nature. This is just computer simulation of kind of those two processes. And you can see like you have, for instance, in the blue one, you count the number of plasmids, and it starts increasing, it gets to a certain point, and then the cell divides. You split randomly the plasmid within your daughter cells, and you keep on doing it afterwards. Of course, there's a probability, like in this orange one, that the cell suddenly loses that plasmid. And the moment you lose the plasmid, you can't replicate because you have nothing to replicate, no? So that's what's called a segregational loss. From a perspective of a medic, that's a good thing because you're losing the resistance gene. From a perspective of engineering, that's a bad thing because you want them to carry. So a lot of people have studied it. So just to see how this segregational dynamics or segregational drift occurs, let's imagine in this cartoon that we have one cell, and it divides and it divides and it divides and divides exponentially, no? And you have many generations. And at some moment in time, you have one mutation occurring in one of the plasmids, and one gene that's carried on the plasmids. So the first thing to notice is that the mutation occurs randomly, as Lurie and Delbruck showed, but it occurs in one copy of the plasmid. It doesn't occur in all the copies of the plasmid at the same time. So then what's interesting, or what is to study what happens with that mutation in this lineage. How does it propagates? I mean, if it does any. So if we have two copies of a plasmid and we assume that the mutation occurred at this moment in time, then half of the cells are carrying the plasmid, and then it starts dividing, and you can see how the redder it is, more plasmid, more mutants. I mean, you have the same number of plasmids, but you'll have the plasmid fraction of the mutants is larger. It's actually mutant fraction. So in this case, you only have the mutants in this particular case, but in some cases, you still have plenty of the parental plasmids. So if we used selective media as Lurie and Delbruck did, and we suddenly select for those cells that are carrying the mutant plasmid, you can see that, well, the red ones are the ones that survive that fluctuation assay. Yes? The mutation? In particular in this drug resistance gene we're interested in. In the drug resistance gene, it's a TEM1 mutation. So the system I'm going to be working on has TEM1 gets resistant to ampicillin, but one mutation away you have lower resistance to ampicillin, but higher resistance or higher affinity to other antibiotics, in particular ceftazidime. Not yet. So exactly. Well, in this case, we aren't using antibiotics. Well, this is just a kind of like a thought experiment. So we introduce antibiotics here and see how many of them survive. So of course, this means that that mutation is dominant, because just having one copy of the plasmid carrying the resistance gene, the resistance mutation, will allow the whole cell to survive. But of course, that's probably not how it occurs. I mean, it could occur on the other case that you'll need all of the plasmids to be of the mutant gene in order to express that phenotype, the resistance phenotype. And if you have those two extremes, you can think of some sort of like they call it the H, the dominance, and you can see how depending on the dominance of that mutation, you can see how many of those cells survive. And the mutant frequency, of course, decreases as the mutation becomes more recessive. So this is with 2 copies of the plasmid. If you have more than 2 copies of the plasmid as you start increasing, first, this is interesting. This is what, in the context of Bonhoeffer, would be phenotypic delay. Because here, all the cells of this lineage have only the mutant plasmid. No? So this is kind of like, it was a time to fixation for that mutation within that plasmid population at the level of single cells to the moment all of the plasmid are homozygote. And this time delay is what Sebastian and colleagues were calling phenotypic delay. As you start increasing the number of copies of the plasmid, the first thing that occurs is that phenotypic delay is increased. And if you start increasing it even further, then it occurs even further, no? And then also the probability of survival decreases. So just a preliminary result of this kind of like a simple neutral drift model is that the frequency of mutants is negatively correlated with plasmid copy number, positively with the mutation's dominance. Basically, as you start increasing the dominance at different plasmid copy numbers, the mutation frequency increases. And similarly, that the phenotypic delay is an increasing function of the number of copies of the plasmid. Okay, having said that, this is not the only source of delay. I mean, this is some sort of like a population genetics delay of plasmids within a population of bacteria. But there are more sources of delay. And as I'm sure you're aware of, if you have this cartoon of the central dogma of molecular biology, and you start assuming that you have 2 different mutations, 2 different plasmids at the level of genes, and that themselves produce mRNAs of that same type, and they produce the proteins that are in fact what makes the cell resistant to one or the other antibiotic. So this is what I was already kind of like talking to you. So you have 2 different plasmids, you start with the same frequency, and you start going, because they're basically the same plasmid, they have the same origin of replication, everything. So the control number the mechanism of control number, it always adds up to the same number of plasmids. The only thing that changes in time is the fraction between those 2 mutants, between those 2 alleles. And as you start seeing, you can simulate forward and try to produce trajectories of possible plasmid dynamics. But what we're interested in is how does that propagate towards the protein level. One can write down a kind of like a standard gene regulatory model when you take into account, I mean, you have to do some things like normalize with respect to the volume, because this is in terms of units of volume, and this is like plasmid copy numbers per cell, and as the cell increases and everything. I mean, you can go into the details, and you eventually produce something that the concentration of proteins as a function of time. And even though the plasmids were varying in time quite a lot, at the level of proteins, they are quite stable. And there are many reasons for that. Volume is one of them. But also, I mean, this is in the order of tens, this is thousands, this is in the order of 10 to the 5 cells. So basically just averaging noise at this level, at the level of the phenotype or the protein level. Of course, you have parameters. And so it seems, at least from this cartoon model, that phenotype would be robust to temporal variations in plasmid copy number. You can, of course, continue. I mean, I stopped, I wasn't showing you the full simulation, but when the moment this plasmid, just by chance, got lost by segregation, then the plasmid, then the cell only carried one type of plasmids. And in that case, we will say that it became genetically homozygote. I mean, they're only carrying one allele of one type of plasmid. As you can see, then, at the protein level, it takes a while before you're actually observing a phenotypically homozygote cell. This has to do, of course, with, I mean, you're not producing any new proteins, but it takes a while before they degrade. I don't know, sometimes less, sometimes better, but there will definitely be a delay. So this is another source of phenotypic delay. In this case, it is important to note that this mechanism is independent of the numbers of copies of the plasmid, as I was showing you in the previous mechanism of phenotypic delay. Here, it doesn't really matter how many copies you have. The only thing that really matters is how does the whole transcription translation machinery works. So then what I'm basically arguing is that there are like, we can think of diversity, how does it changes in time, or heterogeneity, genetic heterogeneity, how does it change in time, both at the genotypic level and at the phenotypic level. So if we have an experiment and we start a cell with 50% of plasmids of one type and 50% of the mutant type, and we track them through time, the segregation replication processes will produce very high divergence from a genetic level, but it will also produce divergence. I mean, it will also increase in divergence. I mean, in variance at the protein level, but it will be much slower than that. Well, that's what we would like to see. That genetic variability increases faster than phenotypic variability. This is hard to measure, at least at the level of single cells. I mean, all the things that we can think of, they're basically just population level measures of diversity. But in this case, it's something that we can measure in the lab, because we can start the experiment with 50% of each one of those plasmids, and because they're producing a fluorescent protein, we can see the relative fraction of both plasmids, or try to estimate the relative fraction of both plasmids, and see how does it diverge in time. To do so, what we're going to do is use a microfluidic system where we have, well, this is a picture. We have cells that are carrying only one type of the cells, the green one, and also that are carrying the mutant phenotype, or the mutant gene, LATEM1. And those are tagged with a red plasmid. So you have green cells and red cells. But you can also have cells that are carrying both of plasmids at the same time. Those are heterozygote cells, and they're usually like orangey type, no? If we use image analysis or processing algorithms and then try to estimate what the average fluorescent is at any moment in time for each one of those cells, we can plot fluorescent intensity, well, GFP against red, green against red, and each one of those dots corresponds to a single cell. This is just one experiment, but it's interesting because you can see how there are some cells here that are basically just green, but they are not red. And this is a subpopulation of cells that have lost completely the other, like these ones. They are green, but they have lost the red cell. Yeah. No, not yet. I'll get into that. But what we can do first is just to, well, basically any one of those cells, we can compute some sort of relative intensity in order to try to estimate the fraction of those cells, properly normalized and everything, carefully done. But we can get one measure, relative intensity, which probably give us some sort of a proxy of what the fraction of each of those plasmids is within a single cell. And this was just a picture, but we can actually take movies. I mean, with time-lapse movies, we take a picture in 5 minutes or so. And this is just a bright field, and we can see the cells. Well, you can probably count, but they are there. So what this device does, there's like a flow here, and there's a trap around 40 times 50 micrometers, where we trap around 1,000 cells in one single layer, so we can watch them under the microscope at the same focal plane. And but this is the bright field. We can also watch them in the fluorescent channels. So we have GFP, the green, and also the red channels. So we can track at any moment in time what the intensity, the relative intensities of these ones, of each one of those cells are. To do so, what we need to do first is to see where the cells are, what is the cell, and then track those cells through time. So here you can see many. The point is that we're trying to track each one of those cells in time. And when we already have the contour, we can try to estimate how green they are relative to the population level average, or how red they are relative to the, properly normalized. And based on these two quantities, we can try to estimate what the relative intensity is. So they are either more red or more green relative to what you would expect if it was 50-50. Okay, so here it's even harder to see, but we're only tracking some of those cells, the ones that we have like longer time series. This is, of course, induces a survival bias, but that's a different story. And here we have kind of like how the data looks like. So we have number of divisions. We can have also time, but number of divisions is probably a more accurate measure. And what we can see is that try to estimate a fraction of plasmids in terms of a single cell. So we obtain time series of fluorescent intensity. If we look more carefully to some of those examples, you can see that even though we know that this genetic process is inherently stochastic, the fluorescent intensities are basically stable for about, 20 divisions or so. So at least from a phenotypic point of view, they're very stable. The other thing we were interested in, and remember that we're not using antibiotics yet, is actually, if we do, this is without antibiotics, but if you do it with antibiotics, you produce a very similar picture. If you see it from a single cell, then you will of course have variations in the intensity of those two colors. But if you're using a selective agent in favor of, the green plasmid, would you expect to see that each one of those cells will kind of like bias or drift towards the green state or not? I'm asking that as a question, and I know the answer, but I didn't know it before doing the experiments. And it happens that we can't, in a way. We can't see selection at this level, at the level of single cells. At any moment, this is, for instance, the relative fluorescent changes in relative fluorescent intensity as a function of time. And you can see that basically there are the same number of jumps to the right than to the left, or to the green than to the red. So they don't seem to be correlated, even though we select for one of the plasmids. So single cells seem to be phenotypically stable and more or less a random walk. If you look at the populations, by a populations I mean not looking at one single cell through time, but now we're considering 1,000 cells through time or 10,000 cells through time, and we use one of the antibiotics, in this case the red one, then what we see is that there's a shift in the mean, but there's also, this is kind of like deviation from the 50-50, and you can see that if you select for the red plasmid, then the whole population drives towards the red plasmid. Each one of those gray lines is not a single cell, but it's just one of these experiments, a single trap. If you look at a single cell, you wouldn't be able to see this shift. However, if you look at the population level, you can see it. You can use, of course, the other selective agent and then select for the other plasmid. And as you would expect, you see the shift going towards the other direction. So we conclude that selective selection modulates plasmid copy number distributions at the level of populations, not necessarily at the level of single cells. So the reason we're interested in populations is because, I'm trying to take you through this journey of from single cells to populations, because what we're interested in, or the original motivation, was to see what would be the advantage or the evolutionary advantage of presenting genetic diversity. This heterozygosity. And so this is a population dynamics model that I'm not gonna go into detail, but basically you have four subpopulations. That they probably lost the plasmid, so they're plasmid free. Then you have like homozygotes of one type, homozygotes of the other type, and you also have the heterozygous subpopulation. You can get from either of those homozygotes, because we're in our system, at least we're just one single mutation away, and also you can go back to the other ones through segregation or loss. So we can write down a system of ordinary differential equations, and what we're interested in is how does it work? What happens expose them to different concentrations of both antibiotics. So we have in the x-axis, these are called checkerboards or some sort of like two-dimensional drug response curves. So you have increasing concentrations of one antibiotic in one axis and increasing concentrations of the other antibiotic in the other axis. This is, well, in the top you see the model and in the bottom the experimental data. You can see that at just the G1 corresponds to the green plasmid. So if the population only carries the green plasmid, then they're only resistant to ampicillin and they're not resistant to ceftazidime at all. On the other hand, if you have that mutant allele and you're resistant to ceftazidime, you're also partially resistant to ampicillin, but you're clearly more resistant to the other drug. If you have a mix of both, and by mix here like a population level mixture, a co-culture of both of these homozygote strains, and you put them together, I mean, you can see how they grow very well in both environments. I mean, they're like multi-drug resistant. They can grow in ampicillin, they can grow in ceftazidime. And the colors here represent the relative frequencies of each one of those cells. So as you would expect, if you use ampicillin, you select more or less for the green ones. If you use ceftazidime, you select for the red ones. But they're more or less both of them there. So this is a mixed culture. And this is also a mixed culture, what we refer to as HT, heterozygous. So, but a mixed culture equally genetically diverse, but at the level of single cells. So here we have half of the populations carrying one plasmid and half of the populations carrying the other plasmid. Well, in this case, all of the cells are carrying half of the plasmids of one type and half of the plasmids of the other type. No? And there's not a major difference. I mean, they're also kind of like very much drug resistant. So one would expect that, well, it kind of makes sense, I guess. However, if you select, well, I mean, we want to see what happens if you have selection. So the experiment we did was start with, 99% of the ancestral, the parental genotype, and 1% of the mutant. We put them in a and we let them grow very happily for 24 hours or so. And the next day we come back, we take a sample, and we introduce it into new fresh media. And we do that for several days, kind of like an evolutionary experiment approach. We're also going to do something else. I mean, we're going to try to, because we want to see how does selection modulates plasmid frequencies at the level at the population level. So what we're going to do is select for one of the plasmids, for the mutant one, the invasive. But we're going to do it in a very drastic way. So we start without antibiotic, and then we start, the next day at 0.25, a quarter of the minimum inhibitory concentration, and we start doubling every day. So every day they see double antibiotic concentration than the previous day. So you end up having like a very high concentration of antibiotic. It's a very stringent environment. And what happens is, this is the population dynamics model. You start with the green ones at 99% and just 1% with the blue ones. And as you would expect, very quickly you select for the mutant alleles. Of course, the green ones are not allowed to, they cannot grow in this environment, and the blue ones can. So then very quickly after days, the 100% of your cells are mutant. And now we're going to compare it. And remember that they were like these two cases and this case were essentially the same from a multi-drug resistance point of view. But now what we're going to do is introduce, 98% of this type and 2% of this type. So in principle, what we're doing is starting the experiment with the same number of plasmids. The only difference is if they are segregated into two different subpopulations carrying just one of them, or if you have one subpopulation that is carrying both of the alleles at the same time. And we're going to repeat the experiment with increasing concentrations of antibiotic. And what we see is something like this. So because the green one cannot grow, it will be lost. It will get extinct very quickly. And then you have those black yellow dots are the frequency of heterozygous cells in time. And you can see how it increases very quickly. And then just through segregation, just through chance, some of those heterozygote cells have lost that gene, that green plasmid, and they're only blue plasmids. And those are, of course, fitter in this environment. So it will take a while before you get a subpopulation of homozygote cells of the right plasmid, but eventually they will appear and they will fixate, as you would expect here. As you can see, there's also some sort of phenotypic delay here, because it'll take like 4 or 5 days to several days to get to fixation. But interestingly, you have a window, a region in time where you have those heterozygous cells. So here, genetic diversity was lost within 2 days, and here genetic diversity is maintained for longer. This has to have some sort of consequences, or so we would like to think. So now this is the data, and the data, it looks a lot noisier than the model, as it usually does, but it does basically the same thing that we would expect. So these yellow shades are different replicates showing that window of heterozygosity. So multi-copy plasmids promote coexistence between ancestral and mutant alleles, at least for a moment in time. Eventually, everything is lost, and you end up having just one type, the fitter one. But at least for a moment in time, you get like coexistence between both alleles. That sounds like a good idea like a bad idea if you are like, if you are in a constant environment or you're selecting towards a certain specific direction. But we argue that having this window of heterozygosity is probably a good idea if you are in fluctuating environments. It was start with 50-50 of those two types. This is the mixed culture. And what we're going to do is first grow them in LB, just media without selection, and then choose the blue drug, and then the green drug next day, and then the next day the blue one. And then we're going to try to fluctuate selection for both alleles. And we're going to compare it with a population that is solely composed of the heterozygous cells, and we're going to repeat the same protocol. So in this ordinary differential equations model, you can kind of track how does the density of each one of those populations change in time. And you can see that here they grow without selected media, so you have both of them, the green and the blue ones growing. And eventually you use the blue drug, and then you select for the blue ones, and then the green ones go to extinction. So when you use the green antibiotic, then they're not as fit as you would expect. And then when it's again the blue one, everything goes to extinction. So if you start selecting, the blue ones go to fixation and they lose the ability to live in the previous environment or the future environment. While in the heterozygous case, what happens is that even though you select for the blue plasmids, you still have some green plasmids kind of like hitchhiking there in the cells. It's not so easy to get rid of them. And which is probably a bad idea if you're in a constant environment, but it's not such a bad idea if you're in a changing environment, because then you're basically already prepared for future environmental conditions. We also did the experiment, and as you would expect, the green ones were only able to survive in the constant green environment. The blue ones were able to survive in the blue environment. The mixture of both were able to survive more or less in the constant environments, but they were not so good at the only one in I don't know, 6 replicas, I guess, 4 replicas were able to survive. While in the heterozygous case, they were basically always able to survive. So we conclude that intracellular genetic diversity increases the probability of survival in fluctuating environments. So in summary, the first thing is that the genotype phenotype is complex, stochastic, and delay. I'm preaching to the choir here because you already know that. The segregational drift produced by multi-copy plasmids increases phenotypic stability. Phenotypic delay produces periods of time where you have ancestral and mutant alleles coexisting, and that this has an important consequence in terms of the probability of survival in fluctuating environments. And finally, I haven't said it explicitly, but what we're kind of proposing is that multi-copy plasmids can be drivers of evolution by allowing and alleviating evolutionary trade-offs. What I mean by this is that if you have just one copy of the gene and you get that mutation, well, there's this big question about how does innovation arises, and even from the times of Ono, they were thinking of kind of gene duplications would be a way of going through trade-offs. How do you get a new function without losing the previous one? And one way of doing it is just duplicating your genome, and then you have mutations on the other copy without losing the previous one. Well, that certainly it's a good scenario, a possibility. But we're saying that basically multi-copy plasmids are, in a way, effectively doing that, because they have multiple copies of the plasmid, and if you get a mutation in one of them, that doesn't mean that you lose immediately the previous one. So we argue that this could be some sort of mechanism that would alleviate evolutionary trade-offs. And thank you very much.
So what happened to Luria and Delbruck? What is your conclusion about that discussion?
Oh, to them, I don't know. I think they were really brilliant because, and it's one of the, I think probably one of the, I don't know if the first, but it was certainly a very good example of mathematics or statistical thinking was able to contribute into biological questions. I think that Salvador Luria was actually a physicist, and he was, the legend goes that he came up with this explanation, or realizing that fluctuations are not an error, but actually a feature of the system, by watching all his friends losing money at the casino while he was kind of like thinking of these things. So what happened to them? They won a Nobel Prize.
No, but what about the discussion?
If it's, yeah. Well, I mean, now everybody would agree that mutations are random, and it's interesting to think about that question now because we give so many things for granted. I think we're getting to a point where actually we start realizing that not everything is so random as mutations, and there are some sort of either epigenetic or some sort of many different mechanisms that actually would produce almost quasi-Lamarckian adaptation. Yeah.
Thank you for the talk. So I was wondering, when you said that you get mutation in your plasmids, have you identified or even sequenced where the mutations were? For instance, could it be at the origin of replication?
Yeah.
So it could be, of course, in the resistant gene. It could be in the promoter. It could be don't you think you could have different mechanisms if the mutations are a bit different?
Yeah. So, well, in the data I was showing you, this is specifically the single cell data, we actually introduced the mutation. So we know what's going on. But in the evolutionary experiments, and I didn't show all the data here, but we observed many very weird things going on. And we sequenced the whole thing, and of course there were mutations in the control mechanism. And if you count, I don't know, after 30 days, you see those cells are surviving. I mean, 30 days of doubling the drug concentration, it gets to the limit of solubility of the antibiotic. And still there were some populations that were able to survive. So then we sequenced those, and we found out that they had mutations in the origin of replication and the control system mechanism. And you will have actually something around 400 copies of the cell. So one adaptive mechanism, in case of very stringent conditions, is actually to amplify the number of copies of the plasmid, and by doing so, increasing the gene dosage and also the ability to resist. Of course, I mean, those amplifications are very costly, so if you put them in non-selective media, in one day you'll lose the whole thing.
Thank you for your talk. Since I work in cancer, I couldn't help but see some parallels. You have shown that multi-copy plasmids are they increase phenotypic stability and alleviate evolutionary trade-offs. And it seems that cancer cells will want to do the same kind of thing in the face of, variable environment as well. And what we see often in cancer is aneuploidy. So you have many copies of not the whole genome, but certain parts. It could be either chromosome or large, arm level events, or even focal, small amplifications where you have, 3, 4, 10 copies of some particular part of DNA. So I'm wondering whether that is a similar strategies that, I just want to know your thoughts on that, and if you've thought about that.
Yeah, I thought about that, and I actually at some point, I don't know, you probably, I'm gonna ask you the question now. Could we think of plasmids as a form of aneuploidy, kind of almost like an effective aneuploidy?
Well, I was thinking that myself. So it's not a direct parallel. Although in cancer cells you also have sort of kinds of plasmid like exosomes that get ejected, and there's just DNA floating around.
Yeah.
And I'm sure that when they get divided, it gets partitioned into the daughter cells in some way. So, but yeah, certainly copy number alterations are very well known and studied in cancer, and it should be somehow related, especially if one thinks about one view of cancer as being an atavistic process, where the cancer cell sort of reverts to a more primordial state, more like a bacteria.
Yeah.
Anyway, that's just some thoughts.
No, but you're absolutely right. I mean, we've seen in experimental evolution experiments that you have, like, I don't know, huge regions of the genome that get duplicated or 2 or 3 times or something. So you do get like duplications that will have like a benefit, many different benefits. And kind of like what we think, but that's just our working hypothesis or motivation, is that plasmids are intrinsically producing already that gene copy number. I would explain maybe why small plasmids that are not able to transmit horizontally are there in nature. No? I mean, it's not clear why do they exist now, because they can be lost through segregation. They're costly, so if you're not providing a benefit, you will eventually get lost to the population. On the contrary, if they're actually providing a benefit and they're having a beneficial gene, then the chromosome can easily integrate that gene into the cell can integrate that gene into the chromosome, and then also render the plasmid kind of like unnecessary. So there has to be, but small plasmids are ubiquitous; they're everywhere. So there has to be some sort of they have to be driving forces of evolution from a different point of view. And our thought is that has to do with genetic diversity and plasmid copy number variations.
Thank you.
No, thank you.
Is there any other question? No? Well, let's thank
thank you.
Rafael again. Please. Now, just wait a minute. With this, we come to the end of the talks. But later, we have this inauguration of the art exhibit, and also we have a cocktail. And to talk about this art exhibit, we have Manolo Cocho here. He was going to tell you a little bit about it. And he's the coordinator of the art and science program of the center. So please, Manolo.
Hello, everybody. I'm Manolo Cocho, coordinator of Art and Science and Complexity program. And we try to present here different artistic proposals. And today we have an inauguration of one exhibition of one very important Mexican artist. His name is Maribel Portela. The exhibition's name is Dark Matter, Materia Obscura. And at 5:30, we have the inauguration there in the hall in front of the reception. And you can talk with the artist if you want, and I hope you enjoy the exhibition. This exhibition was made specially specific for this space, for the Center of the Complexity. It's a sculpture made with paper. The proposed is installation, and I hope you enjoy this exhibition. Thank you very much.